This week we're doubling up on laboratory exercises. The first of these two exercises will be on microscopes. And so we'll be talking about just basically safe handling, correct handling of the microscope. We'll do a walkthrough of how the microscope is used. And those are basically things that are much better for me to do in person when you guys have microscopes in front of you. So what I'm going to do with this video is going to cover the other things, like kind of like the theoretical underpinnings of microscopy. And so a couple of features here, we'll be talking about the general idea that we've got compound microscopes, which basically means that we look through two different sets of lenses. You've got the lenses over here. It's like a magnifying glass. There's like a little cylinder, and what happens is that you look through here, you look in there, and the path of your vision basically goes down, 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 actually into the microscope. And then it gets reflected off of a mirror, and it goes straight downwards. So there's a mirror like right here, mirror. And this pathway down is going to go through another lens. The lens kind of looks like this. It's going to have like a half lens. That's a glass part. And then it's going to have like a little partial cone. And this part of the lens is going to be varying. This part of the lens is the kind of maybe the most important part of the microscope. Yeah, the thing that separates a really expensive microscope from a really cheap one is the quality of the objective lenses. And in our classroom microscopes, we're going to have objective lenses that kind of flip around one to the other. We've got a 4x scanning objective. It's got kind of like a red band. It's going to short and stubby. And we've got a yellow colored 10x or low power objective. And then we've got a 40x high power objective, sometimes called a high dry. And there's also one additional objective lens. I'll mention it here, but we're not going to use it. It's the oil immersion lens, which is something that we just don't need, and it also requires us to apply oil to whatever it is we're looking at. It basically makes the slides and the microscopes really oily, and I'm not going to bring out the immersion oil for our labs. Okay, so this lens up here, the ones that you actually put your eyes against, is the ocular lens. And so we actually get 2 times magnification. We get magnification from the objective lens, further magnification from the ocular lens. The ocular lens has a magnifying power of 10x. And so this is how we calculate total magnification. And it is the ocular lens magnification times whatever objective lens we happen to be using. Okay. and so if we are using the 4x scanning objective, that would basically make total magnifications equal to 40x, right? Because it's 4 times 10x. If we're using the low power objective, this would give us a total magnification equal to 100x. And if we were using the high dry, we would have total magnification equal to 400x. And using the microscopes, it's always easiest to find things and focus using the lowest magnification available. So we always start there. And once you have something in focus with the scanning objective, then you switch directly up to the low power objective. You don't have to move anything. You know, things are in focus in the 4x magnification, 4x objective, and then we move the 10x objective right into place. And the plane of focus should be about the same. We say these lenses are parafocalized, which basically means that it makes it a lot easier for us to use the microscopes. We don't have to continually focus and unfocus our microscopes in order to see things as we move up to higher magnifications. Now there's a third lens here, and I'm afraid I'm going to have to erase my little easiest because I need to show you what's going on underneath the objective lens. Because underneath the objective lens, this is where you put your slide. So here's our slide. And the slide is going to be having some little thing there that we're going to be looking at. And so, yeah, we're going to be having the light passing across whatever it is we're looking at. It's going to be captured by the objective lens. That image goes up, hits the mirror, and comes out the ocular lens where you're going to be able to look at it. Now, there's actually a third lens system that's underneath the scope, because down here at the very bottom of the scope, we've got the light source. Little light bulb. It's like a light pot, and light basically shines up from there. And what we need to do is we need to focus that light, kind of like a spotlight. And so what's going to happen is we'll have another lens system. Basically, another lens kind of looks like this. It's going to take the light from the light source, and it's going to channel. It's going to channel like a little cone, make it a lot more dense. It's kind of like the magnifying glass that you use to burn a little hole in papers. And so this condenser lens is underneath the stage in the microscope. It's just a hole. There's obviously a hole right here in the microscope stage, and that allows this light to pass through. This is the stage right here. And the light passes through. And this condenser lens is actually really important for making our images look really good. There's this thing that we might get into. It's called Kohler illumination, which basically lets us take maximum advantage of our microscopes. In order to do Kohler illumination, we'll have to take the condenser lens, move it up and down, basically to get maximum illumination. There's another little device down here. It's called the iris diaphragm. And the iris diaphragm basically increases or decreases the size of the hole that's letting light into the condenser lens from the light source. So between adjusting the size of the iris diaphragm, adjusting the height of the condenser, we can make relatively slight adjustments to how good the image is coming through our microscopes. Okay, what I want to do at this point is take a little bit of time to do a little bit of a pep talk that I never really get a chance to do when I instruct students in the laboratory. It has to deal with drawing things. Because, yeah, in the lab book, it says, draw what you see. You know, if the lab book says, draw what you see, and all you see is a bunch of random dots, yeah, I know that's an honest assessment of what you see, but it's really not useful for you later on. What you want to make sure that you do is know when you're looking at something. Make sure you're looking at something. And by something, I mean something important. Make sure you're looking at something before you draw it, okay? There's no point in you drawing whatever random little bit that you see in your microscope, because it might actually be just a little fleck of dust or maybe a scratch in the microscope slide. And if you're drawing a picture of a scratch in the microscope slide, it's not going to help you with your biology class, okay? And so what I recommend that you do is to kind of place exactly what it is that you're looking at. So maybe you're perfectly honest and you're saying, okay, when I look through the microscope, I just see a bunch of random stuff, okay? But it's somewhere in there, there's got to be something that you want to look at, okay? Maybe what you want to focus on are these little guys in here, right? And so what you'd like to do is to say, okay, well, let me just draw for you first the field of view. Okay, so this is a field of view, and I'm looking at this at total magnification is equal to 100x because I've been fully advised that I'm using my low power objective, and because I'm using my low power objective, the total magnification is 100x. Okay. Now what you could do is you could say, well, this little thing here, because I can see it a little bit better, actually kind of looks like this. Maybe it looks like, I don't know, maybe it's got one of these things. It's got, okay. And yeah, if that's the little things that you're seeing under the microscope, then you'll want to draw that. Okay? And maybe the lab manual tells you to label the antennae, the eyes, the mouth, and the legs, and you'll do that basically in your lab book. You'll draw a little arrow and you'll say, well, here's the antennae, singular antenna, plural antennae, eyes, mouth, legs. And no, that's not like anything we'll be looking at today. But basically the idea here is that if you see something that you're going to draw, make sure first that it is something. Make sure we know approximately how big it is relative to the field of view. And then once you've got a pretty good image of what it's supposed to look like, make sure you get the salient parts. Because, yeah, I see way too many students spend a lot of time drawing careful pictures into their lab book without any labels. And if you don't do labels, then you don't know what they are later on. Way too often do I see students drawing pictures into their lab book before they're sure that they're actually looking at something. And that might even be worse. Okay, now let's look at today's agenda. Okay, so what we'll be doing today is we'll be looking at several different kinds of slides. We basically start you off really easy. I know most of you have never used microscopes before, so we're going to be starting you off like the first level in a video game, which is just sort of dead easy, just letting you get used to the controls. Okay, so level 1 in our microscope game is going to look like this. We're going to give you a little letter E that's been printed onto a slide. Okay? And your job is to put it under the microscope slide, get used to pointing the objective lens at the letter E that's under the objective lens, and then use the magnification to see how the letter E looks. All right, spoiler alert. Okay, it's going to look like this. That's right, much bigger. You're not surprised by that. Okay, you're not surprised by the fact it's going to be bigger, but it's going to be inverted. The top goes to the bottom. Bottom goes to the top, left goes to the right, right goes to the left. And this is just the way that the optics are in our microscopes. Everything that you see under the microscope is going to be inverted top to bottom. Okay. Level 1.1 is going to be crossed threads. Okay. There'll be a yellow thread, a green thread, a blue thread, a red thread, and they're going to be crossed. And what you could do is you can use the microscope to locate the crossing points of these threads. You should be able to tell using the microscope which thread is on top, which one is second, which one is third, etc. Okay? And so these are basically just practice levels. I'm not going to ask you any questions about letters E or cross threads on the next test. Okay. Level 2, we're going to be looking at Elodea, which is a water plant. Elodea, sometimes it's called anacris. I think anacris is the official name. Okay. And so anacris kind of looks like this. It's got a central stem. It's got these leaves that are coming off the side. The leaves towards the base, they're perfectly good. They tend to be a little bit thicker. And if you get the leaves that are closest to the tip, these leaves are prime. These are the thinnest, freshest leaves. And those are the ones you want to get. And what we're going to be doing is we'll be doing a preparation called a wet mount. And a wet mount is basically a thing where you take a glass slide, put a little droplet of water on the glass slide, and then we'll be taking whatever it is that we'll be looking at. Maybe this little leaf, this little anacris leaf. You take a little leaf off, okay? You just pull it off. Make sure you only have one because they tend to stack up. Put it in there, and then we're going to be putting a cover slip on top of that. Okay, cover glass. Just take a cover glass. Looks really thin. Just plop it right on, and then you'll have this structure called a wet mount. Okay. Try to make sure you don't get a whole lot of water all over the place. We're not going to stain this one. This one is not going to be stained. We're going to be doing a couple of exercises with this. Once you have your Elodea leaf on the wet mount, you're going to put it under the microscope and make observations. Okay, you'll be drawing this. Okay, so maybe you'll draw the field of view, and then here's the arrow, and then you might see a whole bunch of cells look like this. Okay, so cells. Okay. You don't have to do many detailed observations, and maybe you'll take one cell and kind of draw it out and say, okay, well, here's what a cell looks like. And what's really kind of neat about anacris is that when you get under the scope, you'll see that there are little spots inside. And the spots are going to be moving around. Sometimes all the spots will be moving around pretty quickly. They kind of shift around, and those spots are going to be things for you to identify. And not only that, the entire space inside the cell is going to be more or less filled with cell material. And then what's going to happen after you identify all the spots that you need to identify in the anacris cell, we're going to just pull that cover glass up a little bit, okay, and then add salt. Okay. We've got a salty water solution. I think we've got a 10% NaCl solution that we're going to be introducing. We'll be looking at the same Elodea leaf. This is going to be before salt and then after salt. You're going to be coming back and making observations of how that same cell looked. You don't have to get exactly the same cell. The cells should look more or less exactly the same. Okay, and so the question is, well, what's going to happen? What's going to cause the change that we see between the before salt and the after salt one? And that's going to be a question that we'll be addressing. All right, so again, level 2, done. Level 3 is a little bit harder because we're going to have to apply stains. Level 3. We'll be looking at an onion. Uh, not not just any part of the onion, it's that really thin membrane that you see between the layers of the onion. So if you've ever cooked, that's when you take food and apply heat. You know that in between the layers of an onion, so here's my onion, in between the layers of the onion, you have this little thin membrane. It's like wet paper. And you take just a little square of that wet paper, make a wet mount, just like we did before, only this time, instead of using water, or basically you could use in combination with water, we're going to be using a kind of a red-orange stain. Okay? And it's going to be saffronin. You put your little onion in there, and onion slide in there. You put a cover slip on top of it, and now you've got a wet mount of stained onion skin. And there's a whole list. Okay, remember to do a good drawing, identify all salient parts. Not much point am I telling you what those are because you can find them in your lab book. And we'll be talking about them as well in class. Okay. Onion is going to be our example of a plant cell. Onions are plants after all, and level 4, which is actually not any harder at all than the onion, is going to be some cells from you, your cheek epithelial cells. And you'll get these cheek cells by taking a little wooden stick and kind of gently scraping out the stuff, the little slimy layer that's on the inside of your cheek. Okay, and what happens is that the cheek is going to slough off. If this is the inside of your cheek, so here are your teeth. This is my picture of your mouth, and if you basically take the wooden stick and just kind of gently wipe the wooden stick against the side of your mouth, what's going to happen is that a bunch of cells that are just kind of sloughing off of your cheek epithelium anyway is going to be loaded onto the tip of a slide. You'll be putting that into another wet mount. This time you'll be using methylene blue. Put your cells in there, put a little cover glass on top of it, and you'll have a wet mount of cheek cells, and you'll be drawing that. Okay, again, identifying all the salient parts. Remember this. Remember what these epithelial cells look like, because we're going to be looking for similar cells later on in the semester. Okay, I really don't think there's a whole lot more in that microscope introduction laboratory. And it's a short laboratory, which is why we figure it's an okay thing for me to double up this one with the mitosis laboratory, which is also similarly short, microscope based and highly descriptive. Okay, so let's go ahead and back up and start thinking about the other half of our laboratory, which is mitosis. Okay, largely what we'll be doing with this laboratory is identifying the stages of mitosis. Mostly we'll be worried about how these things look in the context of an onion root tip. Okay, this is a topic that was covered in Module 6. And basically the context here is that mitosis is part of an overall thing that we call the cell cycle, which is an area of interest in biology largely because of its relevance to the issue of cancer and cancer research. Okay. Cell cycles are controlled typically, and when a cell becomes cancerous, when it turns into a tumor cell, then its ability to control its own cell cycles is kind of lost. And so mitosis, in the sense that it's part of a cell cycle, is an area of intense interest. There's a stage in mitosis as well where the chromosomes split. That's metaphase. That's also a little bit out of control. Those chromosomes aren't splitting exactly right in the case of cancer cells. So for a lot of reasons, we're kind of concerned about mitosis. And if you remember the whole cell cycle, the majority of the cell cycle is tied up in this thing called interphase. Okay? And interphase is kind of like the entire cell cycle that we have between one mitosis and the next mitotic division. So this whole thing is going to be interphase. It's broken up into the G1, the S, and the G2 phase. And the other part of the cell cycle is mitosis. Okay, and for our purposes, we'll be breaking mitosis into 4 stages: prophase, metaphase, anaphase, and telophase, abbreviated PMA and T for us. And what we're going to do is just learn what cells look like when they're in interphase, what cells look like when they're in some stage of mitosis. And if they're in some stage of mitosis, what the cells look like when they're in prophase versus what they look like when they're in metaphase, anaphase, and telophase. And yeah, it's a very descriptive laboratory. We're going to be doing this with two kinds of slides. One is kind of like level 1. It's the onion, also known as Allium. Allium is the Latin genus name for onion. And this time we're looking not at the skin, but at the onion root tip in kind of a longitudinal section. I'll draw that out for you. So here's the onion up here. Here's the onion. There's the green parts that grow up. I'm not going to change colors for this. You've got these roots that are growing at the bottom of the onion. So if you look at just one of the root tips, it's going to have these different kinds of cells. Basically, we'll have a lot of cell divisions. Cell divisions are going to be going like crazy. And what happens is that these cells divide, and then they elongate. Okay, and the reason why this root grows out this way is because we have cell divisions, and the cells get longer, and they get longer, and they divide, and they get longer. And so this area of the root tip, if you only look at the very tip of the root tip, we've got an area of active cell divisions. And it's for this reason we'll be looking at a cross section of the root tip, like right there. It's we'll be kind of focusing only on that portion of the root tip. It's going to be lines of cells. It's going to look like bricks in a wall. Okay. But if you look at these cells really carefully, some cells are going to be in interphase. You'll see maybe a couple of nucleoli, just generic gray in the background. Some of these cells are going to be in metaphase. Recognize metaphase really easily because I'll have this like obvious dark mitotic spindle. This will be chromosomes in the middle. And so, yeah, what we'll be doing is we'll be looking at these cells, identifying the 4 different stages of mitosis as well as interphase. Now, the reason why this is level 1, okay, this is kind of important. The reason why it's level 1, and we could actually expect to identify most, if not all, of the cells we see in an onion root tip, is because the way the plant is growing, the way the onion is growing is in this direction, right? So we know that all the cell divisions, when cell divisions are going to be taking place, are going to be separating the cells, one going in the down direction, one going in the up direction. Okay? So in other words, every one of these mitotic cell divisions is aligned for us exactly in a plane that makes it easiest for us to see what's going on, what stage of mitosis they're in. Okay, so we'll be doing this, and when you do your onion, don't take your onion slide back up to the front right away because we'll be using it again just after we cover the whitefish. Okay? Level 2 for us is going to be a whitefish. Not just any random slice from a piece of smoked whitefish. We'll be looking at a whitefish embryo, and specifically this is the blastodisc. So if you take a whitefish egg, the egg's going to look like this. You probably know what it looks like if you've ever been to a sushi restaurant. Now the blastodisc is going to be kind of like a layer of cells that's forming at the very top of the egg. And we take that layer of cells and put it onto a slide, and we stain them. Actually, what we do is we put 9 blastodiscs, 9 little tiny baby fish, onto a slide. We apply stain, and then we cover it with a cover glass. It's fixed. Okay, so these are the slides that are already prepared for you, and we'll be looking for cell divisions in these blastodiscs. Now, the thing about animal cells, though, is that we don't have that same alignment that we see in the onions. In this case, the cell divisions can be taking place in any given direction. We'll have a lot of cells in interphase, of course, but those cells that are in mitosis, they might be dividing with one cell going in one direction, one cell going in the other direction, in exactly the plane where you can see what's going on. But sometimes there's going to be a cell where where one of these cells is going to be coming right towards you. Right? Okay, so, and the other cell is going to be going in the opposite direction. And so if we have a cell that's dividing like that, it's going to be much harder for us to actually tell which stage of mitosis it's in. Prophase tends to meld into metaphase, and the earliest stages of anaphase are kind of hard to tell apart. Telophase and anaphase might be indistinguishable as well. So basically what you have to do with these whitefish plastidisc embryos is basically scan around. Okay? You have to move around from one embryo to another embryo. Okay, well now I'm absolutely sure this is what interphase look like. I'll draw a picture of it and then move on. Now here's a cell that's in metaphase. Okay, it's a perfect cell. I'll draw that one. Okay, here's the mitotic spindle, and there, that would be perfect. And then here's another cell that's in anaphase. And so you'll have to scan from cell to cell in order to find those cells that are in mitosis. All right. Now for the last part of this laboratory, we're going to go back to the onions, and this time what we're going to do is to try to take a census of 50 cells, chosen completely at random. Okay? And the idea here is that these slides represent for us a snapshot. Obviously, the onions and the whitefish that we're looking at, they're on fixed slides. They're not going to divide anymore. They're dead. We just took these cells at a particular stage of wherever they were when we made the slide. So this is kind of like a snapshot of an onion root tip that's been frozen in time. And since we're presuming that the cell divisions are taking place at a pretty rapid pace, after all, this is a growing root tip. The root tip is growing actively, which is why we have so many cell divisions, and that these cell divisions are asynchronous. Okay. Which basically means that they're not all happening with all the metaphases happening at once, prophases. All these cells are doing their own thing on different time scales. And so given that we've got the frozen in time onion slide, and that they're asynchronously doing what they want to do, we can presume that, for example, if we have 10% of the cells in prophase, if 5 out of the 50 or 10% of all the cells that we find in my sample of 50 cells are in prophase, then we infer that cells spend about 10% of their time in prophase. And if the entire cell cycle from beginning to end is going to be overall 20 hours, then 10% of 20 hours, or 2 hours, are going to be spent in prophase out of the 20 hours total in the cell cycle. Okay, and we're going to be doing this for all the different parts of the cell cycle: anaphase, metaphase, interphase, and telophase. Okay. And once we have these data, we're going to be filling out a chart. The chart is going to be filled out as you collect the data, but we're going to be doing a pie chart. And in the pie chart, we'll be making it look as much like a cell cycle as we can. So in other words, if we find that 75% of the cells are in interphase, then we want that to be reflected perfectly with exactly 75%, okay? Because like 3/4 would be 75%. Okay? And if you're a real science nerd, you'll be carrying around a protractor with you and say, well, 75% of 360 degrees is 270 degrees. And so you'll know that this angle is going to be 270 degrees, and you can mark that using your handy dandy protractor. Okay. Am I kidding? What do you think? Anyways, I'll see you in lab.